community. Remember the thing about innate, it's born with it. It goes against any pattern that's non-self. Often has what are called PAMPs, which are just pieces of pathogen or maybe toxins, substances secreted from the pathogen. You really can't completely separate the immune system into nice, discrete, separate bricks, but this is one way of doing it that people teach it. The adaptive system has to be programmed. The adaptive system, the cells, and the term they use is not computer programming, which is I'm using as a metaphor to help you understand, but it has to be activated. The adaptive system contains of the B cells, the T cells, and then of course we have all sorts of those, and the antibodies. Now, these are T lymphocytes, one type. And then the other one, which will be completely different, are the B lymphocytes. They look just alike. I don't know what the noise is. I haven't been able to locate it. They look just alike. Are they agranulocytes or granulocytes? So they appear smooth and they look just alike. The only way you can tell them apart is by what they do in analyzing some of the proteins on their surfaces, which are different. For the T lymphocytes, there are several types. There's a TC. C stands for cytotoxic. Guess what those do? They kill cells. There's a TR. Sometimes people use a big R. They used to be called repressor cells, but now they call them regulatory. So they're regulatory. And what they regulate is what? The immune system. If you don't have good control of your immune system, it's going to attack you. And that's what? An autoimmune disease or autoimmune disorder. They used to call it T repressor cells because they turn off what? Parts of the immune system so it's not attacking us, or to keep it under control. TH is the helper. Okay, helper cell. Help in this case refers to ability to help code/activate another B or T cell. They help with the coding. That's why they call helpers. Now, all of these can secrete chemicals that can also be used for communication. The chemicals we're going to go into later as a group are called cytokines, and this is written right over here. Cytokines. And yes, this time I will ask you to know some different types when we get into that part of those notes. So they communicate by touch, and they communicate by cytokines. B cells, they do not kill anything directly. No direct kill. Period. A B cell would differentiate into a plasma cell. Plasma cell will produce, what did I say those are? Antibodies. So antibodies think B cell. Direct self-kill think T cells. T helper cell, this is where it can get confusing, can help both the T or the B cell because he's a helper. He crosses the border. He goes back and forth, back and forth, back and forth. Now, a couple of things. The lymph system. You know what the lymph system is? It drains all the plasma and the fluid from your tissues, which means broken down pathogens, waste products, cells that are dying, toxins that are released. But it's not like a circulatory system, which is a closed loop. It drains one way and dumps into essentially some of the big blood vessels near your heart. It consists of the lymph ducts, where people have been in years past surprised to realize that some organisms will hide there. It has little forts, or little sentinel areas where you'll have a collection of B and T cells and maybe fixed macrophages. Those are the lymph nodes. Can also, the spleen is considered part of this because it helps make immune cells. And you also have mucosa called MALT, which will have some components we're going to talk about, like secretory antibodies, IgA, and just a minute. So a lot of this relates and or comes from or is dealt with in the lymph system. But where does it originate? Not in the lymph system, will rise from your bone marrow. The T cells will migrate to the thymus. You know where the thymus is? It's right over your heart. Over your heart. Don't confuse it with the gland that's in your throat. That's the thyroid gland. Thymus is about over your heart. T cells are called T cells because they will mature in the thymus. They go to the thymus and then they mature. B cells are really misnamed. They were technically named for the bursa of Fabricus, which is a organ in a chicken that we don't have. We do not have a bursa of Fabricus. It's called a chicken. But I try to use the metaphor, even though they weren't named for that they tend to mature in the bone marrow, rise from the bone marrow, and can mature in the bone marrow. So don't let that confuse you. Please associate B cells with antibodies. Bless you. TC cell can attack completely by itself. Regulatory and TH cells cross the border back and forth. Now I'll talk about another concept because I have to give you some definitions before I can jump in. Antigen is any pattern that causes your immune system to what? Protect you, to attack. Any pattern. And then it gets fine-tuned to the definition. My current example is a car. The whole car, if it was a bacterium, is an antigen. Your body will fight that. But aren't there sub-patterns there? Smaller patterns? They call them epitopes. Epitopes. Epitopes are the patterns that are still antigens. They still cause an immune response, but they're more finely defined. Like the tire would be an epitope. Well, that's an antigen that causes a response. The windshield would be a different pattern, right? So that's a different epitope. Still an antigen that causes the body to attack. Maybe we call it the door. There's another pattern. That's another epitope. Every different pattern is a different what? A different epitope. All epitopes are antigens. And antigens is just the big name for anything that your body responds to. I think about bacteria. Let's move away from the car. The body may respond to the flagella. Okay, that's one epitope. May respond to the peptidoglycan. Different epitope. May respond to the toxins released. Different epitope. May respond to the capsule if it has one. Different epitope. Okay, so that's some thoughts. I'm now going to teach you cell-mediated and humoral immunity. It's going to look complex and interactive, but I'm going to show it to you right now. Then I'm going to go through some other things, and then you're probably going to see it. You're going to see it again. You're going to see it again. I do this in a bit of a story because that I think helps people remember. Is it strictly a football huddle or strictly a relay race? No. That is a picture to help you think of how it can work. How it works. Now, if the coding, remember these cells, the B and T cells, will do nothing to any pathogen if they're not activated. Absolutely not. So, they're cell mediated. That means that involves, in this case, the defining element is the TC cell. I didn't say TH, because TH can do what? It can cross over. We're starting way, way back. Here's a new term, an APC, and we'll erase this in a minute. APC, what is an antigen? Is an antigen presenting cell. An APC is an antigen presenting cell. Guess who can be an antigen presenting cell? What did I say about the dendritic cell? They're underneath your skin. They're underneath your mucosal membranes. They're the first cells that encounter an invader. They will take your immune phagocytosis and break them down into patterns. Breaking them down into patterns, they're breaking them down into antigens and epitopes. What do you think is going to be used to code the next cells to attack? The patterns that they receive from breaking down the invader. Does that make sense? Patterns they get by breaking down the invader, what they present to the next cell to attack anything with this on it. Anything with this on it. Dendritic cells are one. What differentiates and or matures from a from a monocyte is your macrophage. And to a lesser extent, some B cells, but that gets a little bit confusing, but that's also true. These are three of your APC, which are your antigen-presenting cells. Which means they have to do what? They have, at least the first two, have to break down the pathogen. Everybody okay? Here we go. We're going to go into the cycle. It's a process. It's a fun process, and it's what's happening in you all the time. You have an antigen-presenting cell, an APC. I'm going to say it's a dendritic cell. It could be a macrophage. It's going to take in this case, it's going to take in some sort of virus. It's going to come around, bring in the virus. Of course, you know you have the phagosome and you have the lysosome, and if you used to give the phagolysosome, virus is broken up into patterns called epitopes, pieces. So you're going to have vacuoles with pieces of what? The virus. We're going to say this has pieces of capsid. We're going to say this has pieces of, let's say, H or N, the virus. This is going to have a piece of an envelope. The way I teach this is think of these as like flags. And I'm going to draw it two different ways. One way that you see in your book and another way I'll use a metaphor. That's not enough. So far you have some patterns that have been separated out or sorted. In each mammalian cell, you have also what I call flag bases on MHC that is major, because it's important, histocompatibility, histo for tissue, I never spell it as possible, and complex. Major histocompatibility complex, MHC. Give you some history. When they, you know Hollywood says that if you're out in the middle of nowhere and somebody's dying and you can just take a hand off of somebody else or a finger and transplant it over and that person's going to be okay, right? That's not what organs do. Tissue transplants, or even blood transfusions. There is rejection, correct? Well, that is what they first discovered MHC with, major histocompatibility complex. They were doing tissue transplants. And they found out that you can't just take the tissue from one person and plop it down into another person, because the body recognizes their tissue as non-self. So there are patterns on the surface called MHC, and I call them flag bases. Major histocompatibility complexes that say, this isn't part of me. That's not why they have an immune system. There are no cavemen going around transplanting tissue from somebody to somebody else. They didn't have the ability. Their reason for existence had to be something else. They are part of the coding system to help activate the TC and the B cells. The MHC, part of the coding system that will help activate the adaptive immune cells to attack the correct part. That's what they're really there for. Now, let's make it a little bit less esoteric. So, in these vacuoles in your immune cell, you have flag bases of MHC made within an immune cell. They are very selective, because what do we say about epitopes? Each epitope is different. The MHC for capsid will only accept the epitope for capsid. The MHC for that H-glycoprotein will only accept for the H-glycoprotein. They have to match. Well, this is still useless, because how can you code something if everything's inside the cell? There's no communication. Guess what? These are pushed to the surface, and you end up with a presentation of the epitope. The epitope would be the flag, and the MHC is your base. The MHC is naturally part of your immune cell. epitope came from where? In there, the pattern. They don't draw it like this in your book. They draw it like this, which is the MHC, and they put the epitope, it's like a diamond in between. It's not even a diamond, but anyway, like different type of draw. Same thing. This would be your epitope. This, both of these, are your MHC. Well, it's presenting, antigen presenting cell, presenting a pattern. It wants to code another cell to attack what? That pattern. Well, you can have coming up, and this can happen in lymph nodes, two other cells that look just alike, a TC and a T helper. And a T helper. TC cells, I said all cells have these flag bases. They will also have a corresponding MHC, and briefly there can be a touch to help with activation. The T helpers also have what? MHC. They can also have a brief touch. Everybody has a brief touch. And they're also going to start secreting, especially the T helper, chemicals, which are as a group called cytokines, and go into type in a minute, that help with more activation. You are what? Coding this cell to attack what? Anything that has that H. Now, it won't bother the capsid. It won't bother the end protein. It will only bother that part. That part, or recognize that part. These have to be in association. That's why I call it the football point of huddle, at least long enough for all the cytokines to start being produced, for all the interactive HMCs, MHCs, and epitopes, for everything to activate this. Some flus, influenza, cheats. Influenza comes in and it sends off chemicals that tell the TH cell to not stay around what I call the football huddle very long. The TH cell doesn't stay around long enough. TH cell doesn't stay around very long. The TC cell doesn't stay around as long as it could. And you do not get what? Activation. Do not get activation. So for cell mediated, you need the T helper to help with all the coding, to get active coding against the virus. He is now turned on. He is going to kill. He is not going to phagocytize. He is not phagocytic. He's going to go to here, and he kills in two pathways, kills two ways. One's called the perforin granzyme system. The other one is called through a protein called CD95, which literally is on the surface. TC cell is going to kill, or can kill in two ways. Some may kill one way, some may kill another. Well, he's going to go floating across or through your bloodstream, or where he's going to go. We're talking virus. It's probably infected what? Your cell. But what did I say the virus was forced to form on the cell membrane? Glycoproteins. Glycoproteins. H&N. Isn't that a pattern? That's a pattern. He's going to recognize that. When you recognize that, he's going to secrete two enzymes. First, perforin. If you have perforated paper, the paper has what in it? Holes. It has holes in it. So perforin is released from the TC cell first. What's it going to put in the target? Hole. This isn't quite like the MAC complex with the complements, a little bit different. The second enzyme, or actually sometimes they all seem to be simultaneous, they can be simultaneous, is granzyme is going to go inside the hole. It's going to turn on the enzyme system in the target virally infected cell to turn on the enzymes to start apoptosis. What if you just cause that cell to kill itself? Kill itself. And that cell will kill itself. And he's just going to go off. He's going to keep killing. He's going to keep killing and keep killing and keep killing and keep killing and keep killing. It's one of the ways. But he will not attack anything else. If a bacterium comes swimming by, or flagella and Peptidoglycan, he's not going to even look at it. Your innate system will go after that because that's non-self. He hasn't been coded, i.e. activated, to go after it. He wait for it. Now, what did I say T cells are known to go after? Virally infected cells. Virally infected cells. So that's the target. Another way he can kill, a little bit different, we are talking about cell cells. So he can come up, and this involves a touch. The TC cell definitely will be activated with the major histocompatibility complex with the loaded epitope, flag base and flag. But he will also have another set of proteins called the CD95. CD95. And they will touch corresponding proteins on the target. This time it's like back to touching your cell phone. He knows to go after this because he's been coded to attack that target. That tells him what to kill. In this case, he doesn't kill with enzymes. He kills with a touch. These two touch briefly, and like your cell phone, a signal is sent inside the virally infected cell. Same thing happens. The enzymes for apoptosis are made to turn on. What does the cell do? It commits apoptosis, cell suicide. Isn't that neat? I think it's neat. That is, in a nutshell, cell mediated. If I say cell mediated, you think TC cell. You immediately think TC cell. Yes.
What goes in and makes the holes?
The perforin, this is one enzyme. This is a second enzyme. There are two enzymes.
What are the letters next to the
These?
cell? Yeah, those N's.
Those are H and N's for the glycoproteins. H and N glycoproteins.
Oh, okay.
Okay. It's all right. You're supposed to ask.
And the T cell is the only one that secretes the cytosine?
Oh no, all these cells
They
can.
all do?
They all can do it.
Okay.
But the TH cells known more to be involved with it. Yes, they all can. Because there are a lot of chemicals flying around back and forth for communication. So, think what I call the football huddle. Do they have to be perfect? No. Does it seem to actually just happen very quickly? Yes. But that's an idea. This could still be a what? Macrophage. Right? But it's got to be a TC, two ways to kill, will not harm anything else. Everybody okay with this one? I'm going to go to humoral immunity. Don't worry, I will show this to you. I will show this to you again. And I'm closing it. Oh, and then stop clicking. All right. I said TC cells, lymphocytes, tend to go after virally infected cells or sometimes cancer cells because virus can overlap. I said, well, you know those. B cells can go after bacteria, which is sometimes what they're known for. In this case, if this antigen-presenting cell, dendritic cell, or macrophage brings in a bacterium, you can break the bacterium down into what? Other components. You can say flagella. You can say peptidoglycan. You can say fibrate. It's still going to do what? Breaks the big, huge antigen with multi-smaller epitope antigens on it into each specific epitope. Loads to a corresponding major histocompatibility complex. Comes up and loads, and I use a flag, but some people don't. I'm going to say this is, I'm going to say peptidoglycan. which would be your epitope over here. It's not going to, humoral immunity is not the TC cell. Humoral immunity is the B cell. What's your B cell? Is it perfectly this way as a metaphor? No, but it helps people keep them apart. Think of relay race. You have I think it crosses the border, a TH cell. So everybody has that LHC, right? He's going to get a brief touch. He's going to get activated. He's going to run off and go tattletale. But he's going to tattletale to a B cell. Again, you can have brief touches, and what are you secreting as chemicals? Different types of immunity. So cytokines help turn on, etc. B cell is now going to be activated, and as he is activated, he's going to take two cell lines, which are going to relate back to the other. Some are going to stay back as memory cells. Memory cells. The ones that we're going after right now become plasma cells. Plasma cells. Plasma cells have inside of them a large number of Golgi bodies. Many Golgi bodies. Think of it as many factories. Lots and lots of factories. They are all going to turn out in very high number antibodies. Antibodies are only going to attack one thing. If this was the peptidoglycan, those antibodies are only going to respond to what? Peptidoglycan. If you have another antigen-presenting cell, or maybe you have another cell line, because each B cell line is dedicated. This B cell line will not touch anything else. You can activate other B cell lines. Have a TH cell over here. It's activated, but let's say that this epitope is to, let's say, the jaw. It's going to run in tattletale. This B cell is going to do the same thing, memory and plasma. The plasma cells are going to make antibodies that only go after what? The gel. The gel. Then the next B cell line is only going to go after capsule. And the next B cell line is only going to go after the next B cell line they go after pili. And the next B cell, you can even break it down from peptidoglycan to those that go after NAM and those that go after NAC. So you have a poor little innocent bacterium. He's got pili. He's got memory. This guy's loaded. But he's loaded with epitope. So each B cell line is going to cover him like 1,000 cockroaches, going after every different epitope. It's not one, it's hundreds and hundreds and hundreds. Isn't that beautiful? Well, for our side. Now, memory cells simply mean, and you already know this because you're in medicine, that the second time you get an infection, these are already coded. They're going to stay around your body for whatever time period. It could be years, it could be months, it could be multiple years. So the next time you get sick, you don't have to do what? You don't have to activate anything because they've already been coded, activated, and they remember. So the second time you get sick, you are more protected against the infection. Third time you get sick, you're probably even more protected because you got more and more and more B cells activated and waiting in the memory in your tissues. Flip side. First time you get the bee sting, not much happens. Second time you get the bee sting, you're sensitized, which really means the B cells have been activated. That is a pattern. It's not affecting us, and you get severe or you can have allergic responses. So it can be good, it can be bad. Okay. TC cells can do the same thing. They'll set up memory too. They'll set up memory too.
That's the same like if you get a transfusion setting for arthritis.
first
Ooh,
time, it's
yes.
not as bad.
You get, well, I think they even attack the first time because it's so non-self, but
Yeah.
it does get worse and worse. And that gets all the way to, I have a movie I might show you later, where a guy, I had a student who said too, she loved peanut butter, but it started itching in her throat. And she ignored the itching in her throat. The guy said the same thing and was saying from the Mayo Clinic, and it started itching in his throat. And he had just enough time to pick up the phone and dial 911 before he was flat and unconscious on the floor, because he had anaphylactic shock. Yes, he ate it again. Yes, he ate it again. Yes, he ate it again. Maybe you can do that your whole life, some people can, and it's just a lower level. Sometimes it goes through the roof. Careful for that one. Well, we just talked about, let's do one more thing, we'll do this one. We talked about memory cells. Let's talk about how these things mature, and then I'm going to go over antibodies, because antibodies have several effects they can have when they come from the B cell. Couple things. You want to mature your B and T cells. You have T cells in the thymus, B cells in the bone marrow. You don't have a chicken bursa, all right? One thing that happens is called clonal deletion. And the reason I didn't teach this first, because I don't think it makes sense until you know about LHC. If, now this is an if you clonally delete correctly any of the B or T cells that one attacks self are deleted. That means the great-great-great-grandparent is destroyed, so there can be nothing that comes from him that could ever attack self. If this is done correctly, the B cells or the B or T cells that don't have working MHCs are somehow malformed with their MHCs are also destroyed. Why would you need to destroy any lymphocyte line that doesn't have a correct MHC attached? That's how you code. If you can't code correctly, you either A, won't code and nothing's going to be attacked, or B, are going to code incorrectly and you're going to attack self. So both have to be removed. Any cell line that destroys or attacks self, and any cell line that doesn't have that correct MHC, because then you can't get the correct coding. You cannot get the correct coding. Does that make sense? Now a little bit down the line, this is after they are already activated, you also can have something called clonal expansion. This actually has to do with one of those cytokines released and others. I want to give you one. This cytokine is called, well, there's other names, but we're going to call you growth factor. Growth in microbiology means increase in cell number, not increase in size. So if you're fighting a battle, wouldn't you want the immune cells that are in the battle to secrete cytokines that cause all the immune cells to increase their replication, so you have more immune cells fighting than fewer? You'd rather have 10,000 fighting, wouldn't you, than 20. So that's what that does. That's what one of the cytokines does. It increases the number of immune cells that they've been activated, just makes more and more and more and more of them. Now, there's another side to this. We'll talk about the B cell again. B cell goes to memory. We talked about better protection. B cell goes to plasma cell, massive production through the factories called Golgi body to give us antibodies. How long do you think this thing lives? He doesn't live as long as he could because all his energy is going into making antibodies. He's loaded with Golgi bodies. All his life is going into giving you antibodies. Antibodies will only protect you as long as they're in your blood. Even if he does suddenly die out and your immune system is cleaned up, the next time the organism comes in, some of your memory cells will stay memory, but others will differentiate into plasma cells. Plasma cells produce antibodies. Everybody okay with that? All right, now let's talk about antibodies. have a handout. I'm trying to make it easier. Trying to make it a little bit easier for you. Keep it like, pretty much. Is there any questions? Pretty much. And raise your hand if you want. I hand you one, I'm asking you to pass them back, please. And there are 5 classes of antibodies. I'm going to go pretty much for this class over 4 of them. And where they come from? Do they come from the TC cell?
B cell.
The B cell. And if you tell me B cell or plasma cell, you'll be okay with me for either one. But know that the B cell differentiates from the plasma cell, and the plasma cell gives you the actual antibodies. The actual antibodies. First, just structure. Put the frog in the throat. I'll be there. On the other side, we have a diagram of a generic antibody. This is your basic core antibody structure. You have 2 what are called heavy chains, and these are made of protein, and 2 light chains. All that means is 2 are longer, and 1 is 2 are shorter. That's what it means, heavy and light chains. They're held together by a disulfide bond that can come from your amino acid such as cysteine. This area around here, the bend, is called a hinge. This area here, when we're talking about how we want to view it as 2 or as a unit, that's called the FAB region. Always called the FAB. This is called the FC. C stands for constant. So F and then AB, hinge is this little area that bends. FC is the base of the Y, base of the Y. As it says, 4 polypeptides, you have 2 shorter, 2 longer ones. The important thing to remember is this. The FAB is also called the variable region. Variable region. That means you can change the proteins on the end. Why would you change the proteins on the end? Because the cell is activated to attack different patterns. If you're going to attack different patterns, you've got to be able to change the pattern on the end of the antibody. Otherwise, nothing's catching. So guess what always links to the pathogen, or piece of it? The FAB part. The FAB part always is the part that links to the piece of the pathogen or the toxin secreted from the pathogen. Now C stands for constant, so the amino acids in this part of the polypeptide pretty much stay the same all the time. That means whatever they're going to link to will probably stay about the same all the time. They tend to link to the immune cell. They will link to the immune cell. So, FAB is the variable region, the part that can change. And it goes after our links to the pathogen, our pieces. The FC region links can already be part of and already embedded in the immune cell and stay there. You can have similar systems like this that are just stuck on the outside of the immune cell. And of course, they're sitting there stuck up. That's actually just called an antibody. It also contains immune proteins. They're called IG. You know what IG stands for? What's IgG? It stands for immunoglobulin. It can also be released and flowing through your bloodstream. So if somebody says immunoglobulin, think antibody. Think antibody, think immunoglobulin. Okay? Immunoglobulin. That is another term. Immunoglobulin. Okay. So, one more thing I want you to think about what antibodies can do. And touch this briefly. Antibodies link to pathogens, or pieces of pathogen. You can have an antibody that will links to a pathogen. You may have pieces of pathogens or pathogens all over the place where you have a site of infection. They keep linking. They'll link again. They'll link again. These will link. You see what's forming? A huge lattice work or net is forming. And it keeps going, and it keeps going, and it keeps going, and it keeps going. You've probably seen this as agglutination. When you have to have blood typing, you see that graininess appear in that fluid. You're actually seeing visually the antibodies linking to what? The patterns they're supposed to go after. Technically, there's agglutination and precipitation, which are all based on this. It's called the antibody or antigen-antibody complex. Antibody-antigen complex. Some people say antibody-antigen. It's the same thing. Precipitation is thought to be more solid. Agglutinations in fluid. I'm not going to be so fine-tuned. As long as you know, it means this. It means antibodies linking to antigens, so you get this huge lattice work that actually becomes visible. On a plate, it's going to come as a fine white line that you'll see in lab. And when we do the fluid, you'll see it as graininess, agglutination. And if it's smooth like milk, no graininess, no agglutination. If they don't match, what happens? Nothing happens. They just keep floating around. There's no linking. There's no linking. Does that make sense? So, know that, and I'm heading for classes of antibodies now. Classes of antibodies. IgG, IG stands for immunoglobulin. IgE as an elephant. IgD, I'm just going to go over if you have enough. IgA as in apple. IgIL as in mythical monster. Okay. IgG is the major one in your blood. Notice he's the classic diagram shape that I showed you. He'll give you up to 80% of the ones in your blood. He is the one, you may add this, that can cross the placenta. He will cross the placenta and protect who? Protect the baby. How long will the antibody protection last? Only as long as the antibodies are flowing through the child's, infant's blood. They're eventually going to disintegrate. There are no B cells to produce anymore. Even if that child has temporary immunity through IgG, he's going to lose it very soon. There's going to come to another antibody in a minute. What's the flip side? Antibody can cross the placenta. Rh negative mother, Rh positive child. Child has what? Patterns that are non-self. This antibody can cross. What can it do? Attack. So we have to have the Rogram shots after you've been sensitized. Rogram. Okay? Now, IgE, we're going to go into this a little later. Allergic reactions will cause mast cells to activate. A mast cell is another immune cell. It's a huge immune cell. Looks like it has a lot of little black dots in it. The little black dots are insoluble histamine. You ever heard of histamine? As long as it stays in that mast cell, you're okay. But if they are triggered by the IgE, the mast cells, they will take the histamine, solubilize it, in other words, put it into solution, put it into the environment which is your body. What's going to release into your body? Histamine. Inflammatory response. What? Red eyes, runny nose. Excuse me. Red eye, runny nose. Why you go over the counter? You just go get antihistamine. Can also help an eosinophil. Will help activate an eosinophil to help attack that one. So they can also help. They don't have to be bad. They don't have to be bad. Okay, IgA, nickname. I'll put this down. Secretory antibody, which means it gives out as in secretions. Secretory, as in being in all your secretions, antibody. Secretions. Same thing with secretions. Well, one secretion is mom's milk. When mom is breastfeeding, she passes to the child IgA. IgA, as long as it's in the child's body, will protect. If she stops breastfeeding, she has not given B cells, which are the generators. She's only given the antibodies. What happens to the immunity of the child? Starts to go away. Well, what you're hoping is the immunity of the child's gotten old enough that they're starting to produce their own immune systems. And the mother is supposed to help protect the child till the child's immune system's coming up, and the mother's immune system starts going down. She can't breastfeed the child forever. So, no. I wasn't trying to say anything like that. Okay. Tears, saliva, mucus, respiratory mucus, even in the mucus in your intestines. There are some autoimmune disorders where children are born and they have no IgA in the mucus, in the lungs. They tend to get what type of infections? Respiratory. Others don't have any IgA in their intestines. They tend to get infections in diarrhea a lot. Okay? So watch out for that one. He's a little bit different. He's two Ys that are connected together by a chain called a J chain. They don't have to be horizontal. They can be vertical, they can be diagonal. It's just two smaller Y forms of immune globulins linked together by a J chain. Do I take that? Now here's the huge elephant. Maybe I call him Mastodon. Okay? IgM is a mastodon, or a mammoth. He is 5 antibody units joined in a circle by a J chain. And one of the things he does is he helps start the complement cascade system. Well, don't you want complement takes time, right? 1 to 2, 2 to 3, 3 to 4. You have all these reactions. So he helps start complement, which of course will help with inflammation and targeting and everything else. When you first get infected, you tend to produce IgM first. And then, of course, with some variations in there, you tend to switch over class switching, it's called class switching, to producing IgG. Why would you want to produce IgM first? Because it takes some time to turn on the complement system. And you want that up in like top. Does that make sense? Then he switches to IgG. And you definitely need that IgA in your mucous membranes. Now, we have talked about antibodies. Let's talk about how they kill. How they kill. Let me even hand out. With antibodies, how they kill. Oh, I thought you wanted it. Oh, no. Oh, good dear. Oh, don't start. Okay, 3. I'm telling you, if you scratch your head in my class, I'll think you're asking me a question. I'm not trying to do that to you. How antibodies kill. How antibodies kill. And on the back of this one is the lymphatic system that we talked about. Appendix. And the MALT is the tissues of all your mucous membranes, your vessels, your nose, your spleen. You already know that. Everybody got one? I'm going to get this frog out of my throat, or you may just have to listen to this sort of deep voice. I put it in the center. In the center. 6 ways they kill. And then I tried to make it like a concept map, which means if you go like a semi-view around the center, that's 1, 2, 3, 4, 5, 6. To show you how antibodies kill. First one is on the upper left, activates complement. That's your IgM, and others can do it too. Right? The complement's going to start the inflammation system. Did we talk about that before? Dilated blood vessels, more fluid into the tissues, redness, etc. I said with an antibody, the Fc region is the part that binds to the immune cell. So the Fc part of the what? antibody binds to the first complement, which is part of the immune system, to turn it on. It then goes in like a domino effect. You start complement protein 1, complement protein 1 starts 2, starts 3, starts 4, starts 5. And if you start the first one, like dominoes, they all go. That's not the first one. That's why it's called the cascade. Cascade, no, it's the domino cascade. Neutralization. I said that toxins can be exotoxins that are small proteinaceous units that are secreted by the bacterium that can flow through your whole body and make you sick. Very small. They link to your cells. They can cause cytotoxic, or cytotoxins break your cells. Neurotoxins hit the nerves. Enterotoxins hit the intestines. Well, what if the Y, top of the Y part, FAB, the antibody, has been fleshy, coated through the B cell, and it links all around the toxin? Essentially, the toxin has been duct taped. It has no open receptors there. If it has no way to link to your cells, it just bounces off and goes on and nothing happens. Nothing happens. So neutralization. Antibody will block adhesion to the surface. Okay? Well, that's what they can block the adhesion molecule on the surface of bacterial virus. That's the next one, on the virus or bacteria. Back to a point of information. For a virus to make you sick, it has to attach. For a toxin to make you sick, it has to attach. For a bacterium even, with the adhesives to, it has to attach. So the antibody can cover up back to the duct tape image. Even on the bacterium, on the virus, cover up the glycoproteins on the adhesives receptors that would match. If you have a layer of duct tape over this, think so, you can't dock. Can't dock. That's called neutralization, both for toxins and in part 2 for the actual pathogen itself. Oh, you're tired of hearing about this one. Oxidization. Remember that one? Iocyan has 2 purposes. Be a flag to mark, like that red dot on a person's forehead because they're about to get shot, which means attack this. Or be a handle or a fish hook so the immune cell can grab it and pull what's on the other end in and phagocytize it, eat it, break it down. So first one says opsonine. Okay, again, you can of course stimulate phagocytosis. It calls neutrophils, macrophages. What do? They bind to the Fc end. What end did I say binds to the immune cell? The Fc. What's on the other end? Draw the picture. I think you already understand this, but I like to draw pictures. If you have a phagocytic neutrophil and you have a bacterium that has a capsule and he's trying to scoot away, all right, but he's been tagged with an antibody. Always the FAB region to the antibody. What is sticking out? The Fc. What is the Fc linked to? The immune cell grabs it. He just pulls it in. He's on the other side of the antibody. You think of it like fishing. He's just going to pull it in. You can think of it like a handle, but it helps him grab it. Helps him grab it. Okay, pulls in pathogen bound to the IgG, and it will, in phagocytosis. Does that make sense? Direct kill, that relates to some other things you heard. When the antibody binds to the pathogen, you may not even need the immune cell. It can be back to touching the surface of your smartphone. It can cause some of this, as right here, production of toxic oxygen byproducts to be overproduced inside the target cell. Well, then we say that you have to have protection against toxic oxygen byproducts. But if they've been overstimulated to be produced in a cell, that cell is probably going to die. They'll break down cell membranes and proteins. Does that make sense? Coming over here, this is the one I already said, agglutination. What is agglutination? It's based on this antibody-antigen complex. Antibody-antigen complex. 2 things of hallucination helps with. Back to what I just drew over here. One, it enhances phagocytosis. How many FC regions are sticking out? A lot. So you can get a lot more grips. Other thing is this is bigger. If you even ignore phagocytosis, since it's larger, it can be filtered out by certain of your immune organs. Maybe your spleen. It filters out better. Flip side. What if all these immune antigen antibody complexes are not phagocytized, which is a goal, that they're not filtered out by your immune organs, which is a goal? What if there's so many of them they lodge in the joints? You get what? What do they say? They say attack. And if these things are lodging in all your joints, you can get arthritis. The immune system is an incredible balance. And that's why you have the TR cells. And also that's why you have the LHC to tell them, don't attack this, because you have to have two things to get an attack. You have to have the correct MHC, and you have to have the epitope. Actually three, because you really need some of the kill the cytokines. That's a lot of controls, because you'll be sure you attack what's attacking you and not attack yourself. All right, here's a funny one. Antibody-dependent cytotoxicity. I said there is a natural killer immune cell. Remember him? NK lymphocyte. NK lymphocyte. Please, don't confuse him with the TC cell before I'm about to tell you right here. Keep the TC cell separate, TC lymphocyte separate from the NK cell. Okay? This one has to have an antibody to know to attack. Has to have a what? Marker to know to attack. Does the TC cell have to have an antibody to know who to attack? No, he's not. He's basing it straight on the epitope on the matrix compatible complex. In this case, antibodies bind all over the pathogen. And what's going to happen here is you'll have all sorts of antibodies binding. We already talked about B cell lines. And then comes an NK killer. The NK killer is going to recognize what side, which part binds to the immune cell. BF, constant, BF, FC. Okay? FC regions sticking out. They're all here. He's going to touch. Okay? And it's going to sound familiar because it's going to trigger the NK cell to produce remember those two enzymes? Perforin and granzyme. And if they're produced, you're going to get holes. Going into the holes, it's the granzyme. And the cell's enzymes for apoptosis are turned on, and the cell will kill the cell. Don't confuse perforin and granzyme from an NK lymphocyte that has been triggered by an antibody with the TC cell that has to have the football huddle with the T helper cell and the antigen presenting cell. Yes, it's the same way of killing with two enzymes, but it's two different control mechanisms. Two different control mechanisms. Does that make sense? Yes?
You said NK had been triggered by antibodies?
Has to be triggered by the antibodies. Has to touch.
And then the other one that's with the other design, the football.
Yeah, the TCs were the football huddle where you have the T helper cell, you have the MHCs, you have the epitope, and you have the cytokines.
So you will see the NKs naturally in what type of, like I'm trying to associate with some type of disease, like to separate this one on here, right?
Well, it can go after a bacterium. It goes after several termites. Okay, none of them are strictly one way. None of them are strictly one. That's just the way they lean. Okay? All right, so now that's the last one. Does everybody have that one? No, I still have time. You know, I'm going to keep going. So, I'm going to switch to the focus notes now. I'll wake up this. some of it is going to sound familiar, you've already heard of it. Some of it I want to try to get. Yes. And usually that. Wake up. Chapter 16, focus notes. Part of it's going to be a walkthrough review, but part of it's going to be new information. Wake up. When I get there. Oh, come on. Where'd you up? Okay, here we go. Couple traits of adaptive. You already know these. Has to be activated, so it's very specific. Okay? Induced means it has to be turned on. To induce means to trigger, to turn on, right? Clonals, that clonal deletion, that clonal expansion. Why do you have deletion? Get rid of the cell if they're going to attack cell. Get the great-great-great-great-grandparent before you have 10 million of them doing it. And also, any that don't code correctly for the LHC, which means they won't have a correct coding system to say due to attack. Expansion means when you do have your activated TC cells or your B cells, you want what? You want more of them to fight for you. Okay, unresponsive cell, that relates back to clonal deletion. That's very bad. You don't want to attack cells. Okay? Memory, for the ones that mean the second time you get the illness, you're more protected because you don't have to go through the APC coding. Memory cells are sitting there, waiting to act. TC cell will go and attack the cell, or some will become active TCs. B cell will go to plasma cell, you'll get antibodies. Not all the memory cells flip to active, some of them stay back. But you gotta keep your memory. And then the second time you get sick, you still make more memory cells. Okay, specific inducible clonal. Lymphocyte simply means to be your T cell. Type of leukocyte, that means white blood cell. B cells, I'm not going to make you know the names for the chicken. T cell thymus. They found this out. T cells need to mature in the thymus. Some children who have heart surgery at a very early age have developed a certain type of problem. Can you tell me what? Because you have the information. When they go in and do heart surgery, what's right over the heart? The thymus. What do they destroy? Sometimes going in to save the child with the heart. But what can't mature correctly now? So some of those children are developing autoimmune disorders, depending on the timing and the age. Okay? Well, that's something that has been happening. Humoral means B cells. The way they're thinking is they're saying antibodies are soluble, so they think soluble means humoral, and that's just the way the names are. I don't see it, but that's why they named it that way. Cell mediated is TC. Who's in both? The T, the TH. Now the lymph system, I want to show you one more thing because I want to draw some more. I like drawing. This is something I'd rather draw. I've got time. When they come back and they know it. We'll see what it is. We're going to talk about a fine-tuned point in regard to the human. Fine-tuned point in regard to the human. I need a column to get my lights in here. Football huddle are loosely called a relay race. Which one is humoral? Humoral is the B cell, APC, okay? APC, which goes to T helper, that goes to B, right? Isn't that what we just did? Of course, the B can go to plasma and go to antibody. That is called, because this is present, T-dependent, of course, it's humoral. Humoral immunity. Because it's dependent on what being here? The T helper cell, so it crosses the borders. There is a little bit of a strange thing about antigens. And this can actually sometimes have they can sometimes conflict with their abilities. The bigger the antigen, usually the more the immune response. Well, what's the flip side? The smaller the antigen, the less the immune response. The more complex the antigen, usually you have more epitopes, the more the immune response. The simpler, more generic, usually the less the immune response. The B cell has one ability. This is T dependent. It can also do T, guess what? Independent. And it's humoral immunity. That means he's still going to have a B cell activated as a plasma cell. Plasma cell will produce antibodies that will attack only that target. But in this case, if the antigens are huge, large, or if they are sometimes very complex, the antigen itself can directly turn on the B cell without who there? The TH. Therefore, it's called T independent. Does that make sense? T independent means the antigen can directly code, if it's very large and very complex, can directly code the B cell to be activated, to attack whatever touched him, and he'll make antibodies against that epitope. This is not as good as immunity. This is weaker in the long run. We could have better memory and longer and better immunity if we evolved to that TH cell. Very much important to get that one. Okay, I'm going to keep going. No, here, nothing up here. Lymph system we talked about. You know what antigens are. We'll talk about this. There's also antigens being endogenous or exogenous. These are just some definitions. I've got to give you the definition. Exogenous versus endogenous. I'm not sure about that. What does exo mean? Outside. What does endo mean?
Inside.
So you have processing of your antigen both outside and inside. Remember, your toll-like receptors go all the way through. Your nods are only on the inside. You don't have to phagocytize something from the outside. If you have vital parts on the inside, endogenous antigen processing. That would be endogenous, which I'm spelling is better. Endogenous, what? Antigen, what? Processing. Processing means putting the epitope on the MHC. Putting the epitope, jamming it down on that MHC, put the flag in the flag base. But if they're inside, you get the part of the virus from inside, because it's already there. Exogenous means the immune cell had to go up and do what? Phagocytize and bring in from the outside something. So that's some terminology. You sure you have that one? I don't care. That's what? Let's start correctly. Don't have any OUS. OUS. I'm thinking of genesis. As in exogenesis, which is something else with the ghost gene. Shall I teach you that? Okay. I should teach you that. Okay. You know that you have these immune cells? Have y'all heard that one? It's going to cost me later. Go ahead.
Did you say that the flag on the outside was the endogenous or
Okay. Endogenous means the antigen comes from inside the cell. Inside. Exogenous means it has to come from outside the cell. I'm going to waste my time, but I'll pay for it later. There was an event that occurred in World War II. It's called the ghost gene. If you ever get a chance, watch Nova and the ghost gene. This is where my other spelling is coming from. The great-grandparents, probably now, but at that time they did a study, maybe the grandparents, in Norway were facing who? Nazis. Okay. Most of them and the villages were starving. Most people everywhere had very little food. Well, that's history. They went and they did a study, and they found that the grandchildren and the great-grandchildren are affected, goes to the gene, okay, by what happened to their great-grandparents in their DNA. And they're still trying to figure out, how does that work? How would the nutritional level, it's called goes to the gene, how would the nutritional level of your great-grandparent affect some of our DNA expression there? They don't know what they're working on. Technically, it's epigenesis, but I wasn't trying to confuse you. Okay? Does that make sense? I'm out of time. I'll have to do a little bit more on this next time because I went to a story. But I think you've been very patient.